Sodium dodecyl sulfate polyacrylamide gel electrophoresis SDS-PAGE. It is a separation of molecules based on size using polyacrylamide gel as a separating medium and sodium dodecyl sulfate as detergent to neutralize the charge of proteins. It is called SDS-PAGE. Acrylamide. In the process of electrophoresis, the acrylamide forms a very stronger gel which is appropriate for separating both nucleic acids and proteins. The acrylamide in very small amount is cross-linked by a methylene bridge of N' methylene bisacrylamide, which results in the formation of a highly porous, mechanically strong and chemically inert cross-linked gel. Principle: sodium dodecyl sulfate SDS is an anionic detergent. When SDS is added to a solution containing proteins, the detergent molecules bind with the polypeptide of the proteins. Thus, number of SDS molecules bound to the protein is nearly half of the number of amino acid residues in the protein. The SDS protein complex has some net negative charge. Therefore, proteins move towards the anode depending on their size. Mobility of the protein molecules depends on the net charge, size and shape of the molecule. Acrylamide solution in a suitable concentration and in the presence of ammonium persulfate solidifies into a gel. Addition of bisacrylamide into this solution produces cross-linking and a sieving gel is formed. Since the separation of molecules depends on the sieves, the degree of sievings can be controlled by regulating the amount of these chemicals. Procedure: 2 dry clean glass plates are taken and a spacer plate is kept between them along 3 sides. This setup is held together with bulldog clips. Petroleum jelly or 2% agar or silicone grease is spread around the edges of the spacer. This setup is called slider. The separating gel is prepared and poured into the space between the 2 glass plates. Amount of water is poured on top of the gel and the gel is left as such for 30 to 60 minutes. Stacking gel is prepared. Water is removed from the separating gel and the gel is once again washed with stacking gel. Then stacking gel is poured on the separating gel in the slider. A few drops of butanol is added to create an even gel slab. Then the gel is dried by blotting with filter paper. The comb is inserted in the stacking gel. It is allowed for solidification for 30 to 60 minutes. The comb is removed carefully without disturbing the well shape. The slider is fixed into the lower tank. Necessary quantity of running buffer is poured into the tank of electrophoresis apparatus. The protein sample is boiled with SDS buffer for 3 minutes and cooled to room temperature. The denatured protein sample is loaded into the wells at different concentrations. A few wells are loaded with standard markers. Bromophenol blue is added to each well as a marker to locate the molecular movements. The upper tank is filled with upper gel buffer. The electrodes connected to the power pack and 80 voltage current are given for 8 hours. When the samples reach 1 cm above the lower surface of the separating gel, the power is switched off. The slider is removed from the chamber. The gel is carefully removed and kept into the strainer for 2 hours. Then it is washed with a detainer until the background is clear. Clear blue sands can be observed in the gel. They are compared to the standards for confirmation. Applications. SDS-PAGE is used to separate proteins based on their mass and size. It is useful to determine the molecular weight of proteins. It is used to separate the different subunits of complex proteins. This method can resolve thousands of proteins in a biological sample. It is used to measure the molecular weight of the molecules. It is used to estimate the size of the proteins. Used in peptide mapping. It is used to compare the polypeptide composition of different structures. It is used to estimate the purity of proteins. It is used in Western blotting and protein ubiquitination. It is used in HIV test to separate the HIV proteins. Analyzing the size and number of polypeptide subunits to analyze post-translational modifications. Thank you.